Okay, good morning everyone. Welcome to the first course in the semester. My name is Jonathan Lowenstein, and the TA of this course, Itamar, is over there. At some point the projector will work and then we will be able to continue. Okay, I will not use it, and everything will be sent to you as part of the documentation of the course. So I'll start with some administrative issues. So what are the requirements? What are the course requirements? First thing is attendance. This doesn't mean that I'm going to count heads and see who is here and who is not here, but the point is that everything that takes place in class, should be you should be aware of and know, and is part of the course requirements. Okay. Second thing is homework assignments, which you will receive approximately every week. I guess that you will not have 14 homework assignments. Maybe few will be missing, but something on the order of every week. Each homework assignment will be composed of an analytical and a numerical part. Those of you that took the computation and cognition course with me, it's more or less the same idea. And you will be required to hand n-1 out of the n homework assignments. Another thing that is not written here is that we would ask for a volunteer every week to summarize what's going on in class, so that at the end of the year we will be able to send you the notes of everything that took place. And those that will hand out the summary of the course will not need to will not need to submit the TA assignment. So who would be the volunteer for the first week? You are? Okay, so at the end of the week send the summary to Itamar and he will post it. The course is composed of, this is a 3 NAS course, 3 hours for lecture and 1 hour for exercise. The separation between the lecture and the exercise will be flexible. In some weeks you'll have 4 hours of lectures, and some you'll have a few hours of lecture and more hours of exercise. This is depending on the stuff that we are teaching and on other constraints. In addition to these 4 hours, there's another extra TA hour, which takes place when?
Usually it'll be Tuesday at 2 p.m., but this week I'll be out of town, and next week. Next week it won't happen at all. This week the first one will be tomorrow at 4 p.m. And it'll all be in a small room right next door.
Okay. And the idea is that since the audience of this course, the background of the audience of this course is rather heterogeneous, the idea is to help those that need help in anything takes place in class. So if you have questions, if you forgot how to multiply numbers or something like this, then you send an email to Itamar, we don't know how to multiply numbers, please help us, and he'll have a class on number multiplication and stuff like this. So there's no, or if you have problems with MATLAB, Itamar. If you have problems in your personal life, Itamar. Everything, this is an extra hour for everything that bothers you. There will be, hopefully, this class will be recorded. There are several hopes here. One, that the video will be recorded. Second, that the audio will be recorded. Third, that it will take place in most of the weeks. And fourth, that these recordings will be posted in the foreseeable future. So if all these things will take place, then there will be video recording of this class, and in that case you don't need to come. Everything will be recorded. Many weeks from now, I guess you still don't care about it, but just to let you know, there will be an exam. And this is the exam, it's an open notebooks exam. You can bring whatever you want, whatever you want that is written, and so you don't need to memorize anything. And the final grade will be composed of 70% exam and 30% homework assignments. Very similar again to computation and cognition for those of you who took the course. Any questions? Syllabus. The topic of this course is the modeling of networks of neurons, and these are the things that we will discuss. Integration, selectivity, the emergence of selectivity using the ring model, memory, decision making, binocular rivalry, and balanced networks. At this point in time, most of these topics don't mean much to you, but we will go back to them at the end of the course. And there are two goals here. One goal is to explain various phenomena at the level of neural activity, behavior, and computation. One goal of this course, but a goal that I think is more important, at least more important for me, is to provide you with analytical and numerical tools for modeling neuronal networks. This is in particular nonlinear dynamics and stochastic processes. So these are the goals of the course, and you'll be able to judge the course in view of these goals at the end of the year. Any questions? Yes?
The topics you mentioned, this is the order we'll be going through them?
Probably. You prefer a different order?
No, I want to be ready.
So the idea is that there are, okay, so the philosophy, the philosophy is to go from simple things that we can understand at the network level to things that are more higher level cognitive functions. This is one axis. Another is going from starting with dynamics and then going to stochastic processes. So this is the motivation behind this order of topics. More questions? Now, some of you have already seen this movie, which I like very much, and I always start my class by presenting this short movie. I'd like to show it to you. And those that have not seen it, I would like you to guess what you are watching. Now, this is a rather boring part. But soon it will free itself. Meanwhile, you can start guessing what is it that you see here. Yeah, that's the end. we can see it again while we try to guess what is it that you watch here. Any ideas from those that haven't seen it? Come on. Yes.
Some path-finding algorithm that you pass in the beginning and go land.
So it's a simulation of a path-finding algorithm. That would be the idea.
I don't know, so any kind of goal-searching algorithm. I don't know, simulating annealing, whatever. I don't know.
Okay, goal-searching algorithm. So this is a simulation, basically. That would be.
Yeah.
Okay. Any other ideas?
Complex system with I don't know, probably or something with here. And somehow there's a constraint that makes it want to go there.
So this is a nonlinear system or something? So this is something real or simulation? Okay, any other ideas? Okay, I'll show you two paths, or two results of two such things. This is one thing, the color, the circles correspond to location and the color correspond to time. So it started here and ended here. And this is a different example where there was some going back and going one way and then coming back, both here and here, but eventually the system started here and ended here. Does it help you? Any other ideas? Yes?
I guess some simulation that tries to simulate the brain or something that thinks and tries to figure out. Says maybe cost function says how close am I right now and where should I go. There is a heuristic that tells you what to do. And it's learning.
So it's idea that is similar to, what's your name? Sorry?
Daniel.
That's idea that is close to what Daniel has suggested. This is a simulation of an algorithm that is tries to find a way from this point to this point, right? That is.
Yeah.
Any other ideas? Yes.
The system with some forces that influence the time and location and pushes to some.
But pushes what? I'll add some more information. So this is the spatial scale.
It's not a simulation.
Well, yeah. Well, I think that you could have guessed that it's not a simulation from the quality of the movie. I mean, if it would have been a simulation, then you could have generated a higher quality movie. You don't need to really take the pictures. So it turns out that this is indeed some stupid nonlinear system. This is the title of this paper is Maze Solving by Chemotactic Droplets. The idea here, basically, this is a pipette. You put a droplet here, and it just there's a gradient of a chemical from here to here, and this droplet moves over on the surface of this liquid, finding its way from this point to this point. So from our point of view, this is a pretty boring thing. I mean, this has nothing to do with the brain. It has nothing to do with algorithms. It has nothing to do with anything that I think the group here would consider as interesting.
What causes the changes between the two ones that you showed us? Like the one that did the half back and forth?
These two, I don't know. These are just two repetitions of the experiment. So on one hand, this is a pretty boring thing. But I think it conveys an important point that we should keep in mind, that behaviors that seem complicated, sophisticated, intelligent, in some cases, these behaviors can be understood as resulting from very simple principles. This, I think, is an important lesson that we should all remember when trying to understand the brain. We should aim at explaining things using simple principles. And sometimes, as in this case, we may be successful at that. So with this introduction, I will start with another introduction, and this is 5 minutes about the brain for those that have no background about the brain. So those that have already taken all the courses in all the ELSE courses on biology, they can play with their smartphones or something. So I'll start with the brain. So the brain is composed of neurons. These are these colorful things. Pretty nice. This is one example of neurons in the brain in a place called, in a brain region called the dendrite gyrus. This is another example from a particularly beautiful brain region. This is the cerebellum. These beautiful cells are the Purkinje cells. These are the granular cells. So the brain is composed of neurons. I think all of you know that. And in this course, we will think about neurons as the basic unit of computation. Now, one can ask the question, why is the neuron the basic unit of computation? Maybe it should be half a neuron, or maybe two neurons together. And actually, this was a question that was of a great interest in, until the beginning of the 20th century. So these two prominent scientists, Gerlach and Camillo Golgi, they supported a different theory that was called the reticular theory. And the idea behind their theory that the nervous system consists of processes of contiguous cells that are fused to create a massive meshed network. Now, these ideas, if you look at the history of neuroscience, they go back at least to the time of Descartes. And then the idea at the time was that the brain is composed of tubes. And when we think about something, there are spirits, some form of animal spirits that move through these tubes throughout the brain. And when they move through these tubes, they change the tubes. And this change of tubes is what memory is. So there was an idea that this brain is just like a complicated mesh network, and the individual elements are not very important. So here we are talking about the end of the 19th century. But at the beginning of the 20th century, there were other people, in particular Ramon y Cajal, and they hypothesized that the brain is made of individual units, cells, and these neurons are cells that are similar to cells in other tissues in the body. And what people saw under the microscope, these nerve fibers are actually cell processes, and that at the microscopic level, nerve cells are contacted. There are sites of contact, but there's no continuity. That are really individual elements. And this was a hypothesis for quite some time, because at the time they were unable, the resolution of the microscope was not high enough to actually view these contact points and determine whether there is a continuum or whether there are individual elements. And of course, today we know that Ramon y Cajal was right and that Golgi was wrong. Both of them, by the way, received the Nobel Prize at the same year. Golgi for developing the Golgi method for staining neurons, and Ramon y Cajal for using this method to identify neurons. And I think many of you know the beautiful drawings of Ramon y Cajal of nerve cells. And today we know that indeed the brain is composed of individual units, and there are many different types of cells. What you see here are just some, not all, some of the nerve types in the retina. And there are many types. There are photoreceptors, there are horizontal cells, bipolar cells, and so on and so forth. And there are ganglion cells that project to the brain, and there are many types of ganglion cells. So we have to remember that these elements that we call neurons are very complicated elements, and there are many types of neurons that differ in the physiology, differ in their anatomy. However, in this course, this is how we will treat all our neurons, as just as a single entity, a simple single entity. But while this is how we are going to treat our neurons in this course, you have to remember that this is how neurons really look like. And in many cases, the heterogeneity and the anatomy and the differential physiology of these neurons are things that are important for brain function. So neurons, this is the first element that we will use in this course. The second thing that we need to remember is that neurons are electrically active. Actually, the development or understanding of electricity goes together with understanding of nerve cells. What you see here, this is a drawing of an experiment done by Luigi Galvani in the 18th century. Do you know what this is? A frog. And what are these wires? Well, the idea is to hope for lightning, and then we'll have electricity going and moving the legs of this frog. And at some point, people thought about some kind of electricity that is specific to animals. And there were big arguments at the time whether there's electricity that underlies what life is all about, rather than electricity that is more general. There were some notions of electricity back from the times of the Greek. But development of or understanding electricity and understanding the nervous system, at some point they developed together, and the nervous system was used actually as a tool to study electricity. And today we know that neurons are electrically active. So this is just an example of a recording of the membrane potential of a Purkinje cell. This is a Purkinje cell from the drawing of Ramon y Cajal, the guy from 2 slides ago. And when you put an electrode inside a neuron and you compare the potential between the inner side and the outer side of the cell, you see these changes. By the way, why neurons are electrically active? Why is there a membrane potential? So neurons, in some sense, are like batteries. There's a difference in ion concentration inside and the outside, and ionic conductance centers, and these are what generate the electrical activity of of these neurons, and changes in the conductances result in changes in the membrane potential, and this is what generates this electrical activity that you see here. This is an example from Purkinje cell. This is an example from pyramidal cells, I think, in the cortex, and this is an example from a Golgi cell, the same Golgi of 4 slides ago. So you see the substantial electrical activity, and common to all these neurons are these spikes that, again, those that the LSEC students know all about. These are the action potentials, sharp increases in the membrane potential followed by hyperpolarization, and then another one. And there are many types of action potentials. Different neurons exhibit different types of action potentials. Those that were in the Engedi retreat know how important the shape of the action potential for computation from the talk of Fred Wolf. But from our point of view, in this course, those that are interested in how these action potentials are generated, those that are interested in how these changes in activity of the cell are generated, can take the course of Tohar on neurons that takes place in the first semester. But from our point of view, we will consider all these events as one type of event, as an action potential. And the action potential is the basic unit of computation. It's a basic unit of computation because, well, first this is something that is stereotypic. If we go back and look at these, all these events are more or less the same. There's not much of a difference between these events. And second, excuse me, to the first approximation, the only information that is transmittable from one neuron to the next is the occurrence of an action potential. So the only thing that neurons know about is the occurrence of an action potential, and that's why we say that the action potential is a basic unit of communication or transmission of information in the brain. So how do neurons communicate in the brain? This is another ingredient that those that don't have any background in neuroscience need to know. Well, neurons are connected through synapses. And the idea is that an action potential that propagates in an axon of one neuron reaches the presynaptic terminal, reaches the synapse, and the result of this electrical activity is a release of small vesicles, these things, with a neurotransmitter that change the electrical activity of the postsynaptic cell. And they can do it either by opening depolarizing conductances, for example, sodium conductance, and then we'll call the synapse excitatory. Or they can do it by opening hyperpolarizing conductances, for example, chloride conductance, and then they generate what is known as an IPSP, hyperpolarization of the postsynaptic cell. So this is the main form in which an action potential in one cell affects the activity or changes the activity of the postsynaptic cell. This is how information or activity in one cell is transmitted to another cell. So these two synapses, these two types of synapses are called chemical synapses, and there's another third type of a synapse that is called an electrical synapse. An electrical synapse actually is very close to the reticular theories from the 19th century, and basically two neurons touch each other such that ions in one cell can move through these holes to the postsynaptic cell. These are called electrical synapses or gap junctions. And as a result, if there is some depolarization in one cell, this depolarization through the passage of currents through these gap junctions causes a depolarization in the postsynaptic cell, and the same thing for hyperpolarizing currents. Any questions so far? I know that this is trivial to those that have some background in neuroscience, but still, I'd like to touch bases. Any questions? How is this synaptic input integrated? Well, in the central nervous system, neurons receive thousands of synaptic inputs. And the postsynaptic neuron integrates all these signals and, in quotes, decides whether or not to generate an action potential. And we can think about spatial summation. If you have EPSPs or activation of synapses in different locations along the dendrite, they sum up together to generate a larger response in the memory potential. And if this response is sufficiently high, it will generate this stereotypic event that we called an action potential. There's an important temporal aspect to it. These events have to occur at approximately the same time, because after each event, each of these EPSPs has some temporal duration, and integration can take place only if these events take place in temporal proximity. Yes?
What's the time scale of the integration?
So the time scale of integration is between milliseconds and tens of milliseconds. However, I mean, let's say that this is to a first approximation. When people who study these things tell us that there are multiple timescales involved in these processes, and this idea that we have, some people would argue that this idea that we can have what physicists would call separation of timescale is wrong. But still, this is what we will do. More questions? So you've already seen that the brain is pretty complicated. You have different types of neurons, and you have different types of synapses, and it's all very complicated, and we have to worry about space, and we have to worry about time. And the situation is even worse, because we have something like 10 to the 11 of such neurons, and 10 to the 14 such connections. So the situation is pretty bad, to be honest. So we need to make some approximations. So the first approximation was to consider all neurons as if they were just one entity. A second approximation was to consider action potentials and not worry about the exact shape of the action potential. A third approximation was to simplify this very complicated machinery that is called a synapse. People can spend a lifetime, and many people have spent a lifetime studying these very complex machinery, the synapses, but we will not worry much about them, and we will use a very poor and abstract approximation to what a synapse is. There's another very interesting observation when we look at the brain, and we will use it at this point in the course. We will use it as an excuse to working less hard, to further abstracting our view of the brain. Later on, towards the end of the course, we will try to understand this phenomenon. And the idea is that, well, these action potentials, the timing of the action potentials, are pretty irregular. Let's go back to these action potentials. So what you see here is the response of a neuron to the injection of depolarizing current at different amplitudes. So small amplitude, medium amplitude, high amplitude, and then you get very regular, you get action potential at a pretty regular timing at a low frequency, intermediate frequency, and high frequency. So this is all very nice, but this is not what we see in the brain. So when we look at the activity of neurons in the brain, in particular, when I say brain, I mean cortex. In other brain regions, it can be different, but in the cortex, the region that we think is more important for high-level computation, this is what we see. So what you see here is a very old experiment from 1 million years ago, approximately. And this is in this experiment, a monkey was trained to memorize a particular shape in a delayed comparison task. And what you see here, these black things are the timing of action potentials. This is approximately 15 or 16 seconds. And and these are repetitions of the same experiment. So one thing that is horrible, especially for the scientists among you, is that you do an experiment and, well, you cannot repeat it. The results don't repeat. So this is one experiment, this is a second experiment, a third experiment, same neuron, same image that the monkey was trained to memorize, but we never get the same results. I mean, imagine that you do an experiment in chemistry or in physics, and you will try and publish such a thing. We did an experiment 10 times, every time we got a different result. This is what we have, right? But this is what we find in the brain. So the timing of events are very irregular, and this is not only because this is a shitty experimental group. This is what everyone finds. This is another example. This is from a very influential paper by Georgopoulos. Again, many, many years ago, in this experiment, a monkey was trained to move a manipulandum to one of 8 directions. Again, these are called, to those that don't know, these are called raster plots. These are the timing of action potentials in a particular neuron in the primary motor cortex. You repeat the experiment 1, 2, 3, 4, 5 times. Well, and you get something. One thing that you get is that whenever the monkey moves its hand in this general direction, the neuron fires more spikes, there are more action potentials. Compared, for example, to the case where the monkey moves its hand in this direction. 0, I think is the time of the instruction to move the hand. So this is one. This is the time of initiation of movement. So you see, there are fewer spikes here than there are here, but spikes are irregular and they don't exactly repeat. We are unable to repeat the experiment. For the same condition, same monkey, same neuron, you get slightly different timing of action potentials, and as a result, at least from an experimental point of view, at least from an operational point of view, what people do is consider only the firing rate, the number of spikes in some unit of time, average over all these repetitions. So when the monkey, the way that that Georgopoulos summarized these results was by saying that whenever the monkey moved its hand in 150 degrees, this is the number of spikes per second that the neuron fired, whereas when it moved its hand in 45 degrees, this is the number of spikes the neuron fired in 1 second, and so on and so forth. So not to worry. So in this experiment, people decided from an operational point of view not to worry much about the exact timing of individual action potentials, but rather to consider only the firing rate, only the average number of spikes, average both in small units of time, but also in ensemble average over, perhaps ensemble average over repetition of the experiment. Just to show you that this is not because This was done many, many years ago. This is a newer experiment. This is in a mouse that is presented with visual stimuli of different orientations and different contrast. And again, the different lines are different repetitions of the experiment. These are after plots, and these black points are timing of action potentials. And you can see that the firing rate increases for this contrast, perhaps 100%, and for this orientation, perhaps 270 or 300 degrees. But still, firing is pretty irregular. I'm zooming in on some of these, and you see that firing is irregular, and the way that people treat these experiments is by averaging both over trials and averaging over time, and by presenting us something like firing rate as a function of contrast, firing rate as a function of orientation, and so on. So people tend not to worry too much about the timing of individual spikes, and so we will follow, when trying to model the brain, we will follow the same approach, and at least in this part of the course, not worry about individual spikes, but rather worry about a measure of general activity, perhaps average firing rate of the neurons. Any questions? So if there are no questions, I will start by writing down the equations that we will use, that can be thought of as perhaps as standard equations for studying brain activity. So what I'm going to write down now is perhaps the most important equation that I will write throughout this course. And we will use it again and again and again when modeling different things in the brain. So far you've all learned that there are neurons in the brain, and that we care about neurons, and that there are action potentials in the brain. And at this point in time, we don't care about the timing of individual action potential. Rather, we care about some more general measure of activity, which can be thought of as perhaps the firing rate of the neurons, how many spikes the neurons fire in a particular point in time, average over many repetitions. So we will denote by SI the firing rate of a neuron. So this would be the firing rate of neuron number I. And we will argue that this firing rate of the neuron is some function of the total input that it receives. So G would be some general function, and HI would correspond to the input to neuron I. Now, going back to the presentation, you will soon see that the firing rate of a neuron is a monotonic function of the input that it receives. So, for example, if you inject depolarizing current to a neuron, it fires at a higher rate than if you inject a higher polarizing current. So this G function would look something like this. there will be some values of H, of input, such that the neuron would be completely quiescent, will not fire any action potentials. There is a saturation. At some point, neurons cannot fire above some threshold, so there will be some saturation to this G function, and there is an intermediate range of values of H such that G increases with H. So this would be the generic shape of this function G, some sigmoidal function that denotes the by how much the firing rate of the neuron increases with the input to the cell. Questions about this equation? The second equation that we need to write down tells us something about how the input to the neuron depends on the activity of all neurons in the network. So this is an equation that will incorporate, for example, everything that we have learned about synapses. So I will write down the equation and try to explain it. The equation is tau hi dot is equal to minus hi plus sum over j of jij fj plus hi 0. This is just the slide that I wanted to show you, showing that the firing rate of the neuron increases with its input. So this would correspond to this equation. Let's look now at the second equation. Tau hi dot is equal to minus hi plus sum over j of jij fj plus hi 0. So let's try and understand this equation. Let's do it after the break. Let's take a break now and we will discuss this equation after the break. So before the break, I wrote down a set of equations that describe a model for how the activity of neurons changes as a result of different things. So, and I didn't fully explain all the terms here, and I will do in a second, but I think we will spend approximately half of the course discussing these equations. And the point would be to see how, depending on the shape of this G function, and more importantly, depending on these Jij's, which I will explain in a second, how different types of computations and dynamics can emerge in the brain. So this is these equations are pretty important. So let me try and explain these equations slowly. So so we have n neurons in the brain. We have n neurons in the brain. Usually we'll think that n is large. Today n will be small. And I is the index of the neuron I. And we will assume in general that all neurons are identical. How do I know that all neurons are identical from these equations? All of them respond to input in exactly the same way. All have exactly the same G function. This G function, we can already guess that it will have some kind, this type of a shape. Because we think about S as a measure of activity, perhaps firing rate, and we know that firing rate cannot be negative, right? And we know that there's a physical limit to the rate of action potentials in the brain. I mean, just a simple way to think about it, an action potential takes something like 1 millisecond. So surely the brain cannot fire at a rate that is larger than 1 kilohertz, right? That's for sure. In practice, cortical neurons fire at a much lower rate. The highest firing the neurons that fire at the highest rate in the brain fire at a few hundred hertz, but typically in the cortex we see several hertz. The firing rate is several spikes per second or several tens of spikes per second. So this tells us something about this function G. And H is the input to the cell. So what are the ingredients to the input to the cell? There is external input. Hi0 is an external input. We have sum over J of Jij Sj. So the Jij corresponds to the strength of the synapse of neuron synapse from neuron J to neuron I. So this is a Jij. So one major abstraction that we did here was to take this very complicated machinery that is called a synapse and replace it by a single number. So this is a very substantial, this is an abstraction, this is an approximation, a very useful approximation, but still we have to bear in mind that this is an approximation. And finally, we have this. This is a differential equation that basically tells us that the input to the cell is a low-pass filter of this input, both the synaptic input and the external input, where this tau would correspond to the time scale, perhaps the synaptic time scale that we were discussing in the previous hour. Perhaps this would be, I don't know, on the order of 10 milliseconds or maybe 50 milliseconds. So this is this would determine the time scale of all the events associated with computation here. Yes. This HI0. This is the external input to neuron number I. So one way of thinking about it, are several ways. I'm hesitant answering your question because you can think about it in a different way. You can think about it as input, for example, from a different brain region that is currently not part of your modeling. That's one possibility. Another possibility is that if you inject a current to this particular neuron in an experiment, this would be the current that you injected. You put an electrode in the neuron, you inject current, so this is the current injected. So there could be different things. But this is, we allow for some external input to the cell that is not part of this network that we are currently modeling. So S corresponds to, is a measure, S is a measure of activity. For example, you can think about S as a firing rate, as you said. Now there is an issue of what are the units, perhaps, of this equation. Now, in order to determine the exact units, we need to be slightly more precise about the model. In particular, we need to think what is it that we measure when we need to think exactly how do we relate these quantities to things that are measured. So we didn't say anything about the units of the Jij. So basically, the units of Jij times the units of S will determine the units of H. And I will be sloppy here, and will not discuss units. The only thing that we need to bear in mind is that computations or neurons typically work in the brain in these timescales of milliseconds. So they're extremely slow. Thinking about it compared to computers, these are many orders of magnitude. Neurons are many orders of magnitude slower than computers. So everything that we do takes in any stage in a processing of information takes a few milliseconds at least. Yes. Perhaps I will. At some point today, I will very soon. I will solve it for the case of Jij is equal to 0, and then perhaps it will become clear what I mean by low-pass filter. More questions? So this is one way of formulating the equations that are of interest to us, equations that explain the dynamics of neurons in the brain. There is an alternative formulation that is not identical, but is almost identical, and sometimes we will use it. I will write it here. Sometimes people refer to it as population dynamics. So the population dynamics equations are tau Si dot is equal to minus Si plus G of Hi, where Hi is equal to sum over J of Jij Sj plus Hi0.
What is this calculation that happened?
So, I will answer it in a second. I'd like to say a few things before that. Well, one thing that perhaps that I should have said about this Jij, I said that Jij is some measure of the strength of the synapse. So we took this complicated machinery of the synapse and we replaced it by a single number. But this number is a measure of how or the extent to which activity in the presynaptic neuron affects activity in the postsynaptic neuron. So if Jij, let's think about Jij that is positive. If Jij is positive, then activity in the presynaptic side will increase H. And if it will increase H, it will increase S. So Jij that is positive is a way of modeling synapses that are excitatory. Similarly, if Jij is negative, then activity in the presynaptic neuron will decrease the value of H, resulting in a decrease in the firing rate of the postsynaptic neuron. So Jij, it's rather straightforward, but still it's a good idea to write it down. Jij is larger than 0, correspond to excitatory synapse. And Jij negative would correspond to an inhibitory synapse. So this is one thing that I wanted to say. Now going back to the difference between this set of equations and this set of equations. In this set of equations, the firing rate of a neuron is an instantaneous function of its input. The time that it takes things to integrate happens at the level of the input to the neuron. So this is what we have in this equation. In these equations, the input is an instantaneous function of the activity of the presynaptic neurons, but it takes time to the neuron to respond to this input. So the difference between these two equations is that the time scale moved from the dynamics of the input to the dynamics of the activity of the neuron. Now, for all practical purposes, the two equations, as I said, are almost identical. They have the same fixed points, the same stability. One way of seeing it is just writing down. I mean, taking these equations and looking at the dynamics of H. So how do we do it? Let's look at tau. Can you see when I write here? Or it's too low? It's okay? So tau Hi dot is just equal to what? Sum over J, Jij Sj dot I forgot the tau. Now I can put this I put this equation, and this is just equal to minus Jij sum over J of Jij Sj plus sum over J, Jij G of Hj, which is just equal to minus Hi plus sum over J, Jij g of hj plus hi0. So basically we get the same equation as before. So these are two equivalent ways of thinking.
How did you derive the first equation?
Which identity?
Yes.
This-
I'm missing the second derivative of.
It's not the second. Just taking the first derivative of h, I have derivative over s. This is constant. Take the temporal derivative of h. Okay, so I have only derivative over s. I assume let's take h to be constant. And then okay. And when modeling brain activity, we will sometimes use this set of equations, and sometimes we'll use this set of equations. But basically,
Why are you calling it population?
because here I'm thinking about this population as a dynamical variable, and here I'm thinking about the synapse as a dynamical variable. But this is name. You can call it whatever name you want. Doesn't matter. Okay, so we have a set of equations, and well, we have two sets of equations, but as I told you, they are very, very similar, and we will soon, or not so soon, see that many interesting things can emerge. Many interesting things can emerge from these dynamical equations. But as a first step, what would be perhaps useful to do is to try and get some intuitive understanding of what's going on and what these equations mean. And this is what we will do next. Any questions? And the way that we will do it, we will consider several examples. And these examples will start easy, and then they will become will become more and more complicated, perhaps more and more interesting. So let's start with the easiest. So the first example. The first example will be isolated cell, or isolated neuron, for which Jij is equal to 0. So basically we only care about this equation for the input, for the input to the neuron, tau hi dot. We can remove the I because all neurons are still exactly the same. Well, they're isolated, so we can just as well consider a single neuron. Tau h dot is equal to minus h plus h0, let's say h0 of t. Let's make it slightly more interesting. There's some more general. So somebody asked here about why is it low pass? This was you. So the easiest way of seeing how this is a low-pass filter is to consider these equations to do a Fourier transform of these equations, of this first-order differential equation, do a Fourier transform, and then this becomes an algebraic equation, and you can see that the high frequencies are attenuated more than the low frequency. That would be the simplest way of doing it. If we do things in the real space, basically the equation of this equation, as you all know, is h of t is equal to h of 0, e to the minus t over tau, plus 1 over tau, integral from 0 to t, dt prime, h0 t prime, e to the minus t prime over tau. So those that don't know how to solve this equation, it's high time that you will learn it. And one possibility, as always, is to send an email to Itamar, and he can do an extra hour on this topic. I'm sorry? But the thing is that you have to send an email in advance to Itamar, because he also has to learn these things. Okay? But seriously, the goal of this extra hour is exactly to address these points that some of you may have missed over the years of your study. So let's just solve it for the case of h0 of t is constant. And then the equation becomes h of t is equal to h of 0 e to the minus t over tau plus h0 e to the minus t over tau times e to the t over tau minus 1, which is just equal to h0 plus h0, well, there are too many zeros here, h0 minus, well, different h0 e to the minus t minus t, e to the minus t over tau. And basically what we have here is that within this equation forgets exponentially fast the initial conditions and converges to the fixed point solution of this equation, h0. That's easy, right? Questions about this? Okay, let's do something slightly more interesting. The second example is something that is known as the OTAPS. What is an OTAPS? An OTAPS is a neuron that innervates itself. How many inputs does a cortical neuron receive, approximately? I'm sorry? It's between 1,000 and 10,000. So, if we think about the cortical neuron, one single synapse that results from, that is, in which the cell innervates itself is not extremely important for understanding the brain. However, it is a rather useful example theoretically when thinking about dynamics of networks. So you can think about it as the simplest network possible, right? It's a network with synapses. Well, not synapses, synapse. It's a network composed of one neuron and one synapse, and we will write down the equations, okay? So the equations are tau h dot is equal to minus h plus j times s. So the activity of the neuron is denoted by S, and the strength of the synapse is J. There will be some external input, H0, which we will assume that it is constant, and we know that S is equal to G of H. So far, so good? Yes?
What is the H0 if we're talking about only the...
Well, again, you can think about it in two ways. Let's say that you're an experimentalist. You put an electrode in this neuron, and you inject current. That's one way. Another way is, let's consider all other inputs that you don't care about at the moment. So basically, let's imagine that this neuron receives also 5,000 inputs from other sources, to which it is not, whose activity is determined by some other processes. And everything will go into this H0. So this is the first network that we encounter. And the question is, how would this network behave? So what are the tools available to us when studying this rather simple, perhaps, network? So what should we do? How should we study this network? What can we do? I'm sorry? Okay. Why is it difficult? Where is it difficult? Where does...
G is unknown.
I'm sorry?
G is unknown.
No, it's not unknown. This is G. Very known. G. Okay? This was measured experimentally. Exactly this. And some error bars. Okay? Well, it's difficult because it's a differential equation, right? And we don't like differential equations. It's ugly, it's misery to our lives. So what can we do? The first thing would be to try and study the fixed point, to analyze the fixed point, right? So what's the fixed point of this equation? Well, the fixed point is very simple. H dot is equal to 0, so the fixed point is H is equal to J times G of H, and G we have from our experimental measurement, plus H0. Right? This is the fixed point equation for this differential equation. Who was unhappy about the G function, not knowing the G function? You were unhappy about the G function. Well, we know now the G function, but now I need to tell you something about J. I need to measure it experimentally, right? And H0. These are the parameters of this equation. I'm sorry?
Yes.
No.
Do we suppose there is constant in time? What's the...
Well, okay, so it's a good point that I should clarify. We will assume that the connectivity in the brain is constant at the time scale of the dynamics. Let me say it again. The time scale in which synapses change is substantially slower, or the time scale is substantially longer than the time scale in which the activity of neurons change. Just to get a feeling, you're in this where you're studying computational neuroscience. Hopefully, within 14 weeks, you will be experts in computational neuroscience, which means that your brain has changed. We believe that what has changed in your brains are the synapses, and it will take your synapses 14 weeks to change in order to make you experts in neuroscience. However, while you study computational neuroscience, the activity of the neurons in your brain will change in timescales of tens of milliseconds, hundreds of milliseconds, and so on. So there's at least when modeling learning, we will typically assume that there's a separation of timescales. Things that happen with the timescale of changing the activity are much shorter than the timescale of learning. So this is one thing. Second question, that you said something about external input. So external input can change while things change. In this model, particularly, we will assume that it is, in this example, we will assume that external input is either constant or changes at timescales, again, that are much longer than this tau timescale, allowing us to analyze this equation as if H0 is constant. Okay, so we go back to this fixed point equation that we need to study, and we already have this G function that we measured experimentally. And what is missing now is that I will tell you something about the value of H0 and the value of J. These are two parameters, and with these two parameters you will be able to solve this equation. So rather than giving you the value of J and the value of H0, the question that I will ask you is, how does the solution depend on the value of J and the value of H0? This would be the question. So how do we go about addressing this question? Yes?
We see what happens with constant negative values of the zero value of J.
Okay, so zero value we already know, right? This is, this we've already solved. This is 0, and we will focus here on J that is positive. Let's add it. Let's just focus on J that is positive because it's more interesting. We will do something with negative J in one of the following examples. So basically what we need to do is we need to find the solution to this equation, right? Solutions. So how do we do that?
If H is very, very small, G is 0, and if H is very large, so G is H multiply some parameter.
So let's what's your name? Peleg. So Peleg says, well, let's consider the case of, let's think about what happens when H0 is very, very small or very, very large.
No, H,
H.
Yeah.
Well, H H is what we're trying to find out.
Yeah, but we want to know how it changes in those limits.
Well, I can't assume anything about H, because H is determined by H0 and J.
But this will allow you to find G, like to simplify G.
Well, but this is not necessarily a relevant regime, because we don't know whether H is well, we don't. The value of H is exactly the solution to this equation. Yes. Let's plot it. Good idea. So what should I plot?
J times G plus H0, there is G by H0 and J and H.
Okay, so let's start. What's your name?
Leon.
Leon. So Leon says, let's plot things. So Leon says, let's plot, well, basically what we need, if we can plot the left-hand side of the equation, we can plot the right-hand side of the equation, and when these two curves intersect, then we know that we have a solution. If they don't intersect, then we are in trouble, but, well, they will intersect. So let's do it. But let's focus first on the left-hand side. Let's on the right-hand, well, left is easy. I think all of us know how to plot the left-hand side. So what I'm going to do is I'm going to plot the right-hand side of the equation. Let's call it right-hand side. This is just too confusing. That's right. J times G of H plus H of 0. This is what I'm going to plot. And I'm going to plot it as a function of H. So this is what I have. So, well, this is just copying this equation for H0 that is very small. So how would this equation change if J is, let's say, much smaller or much larger? It will be stretched. So let's plot it now for so this would be a large J, and the blue would correspond to small j. So this is one way of plotting this equation. And let me delete this one. And now I'm going to plot it again, but now rather than changing j, I'm going to change h0. This is the second parameter. So this is experimental finding as before. I'm plotting j times g of h plus h0. And I'm going to plot it with a different color. I will use it for, this would be for, so what would be the effect of changing h0? This will just move this equation up or down. So this would be large h0 and this would be small h0. Okay, so we started by plotting the right-hand side of this equation. So what would be the next step? Plot the left-hand side. Let's use a different color. So what's the left-hand side of the equation? H is equal to H. That's easy. So this would be H is equal to H. There. I could have. I'm cheating a little bit. But that's okay. I'm allowed to do it. I have tenure. Okay? Let's focus on this side, on this side. So what did we learn? What did we learn from this exercise? Well, we learned several things, except the fact that I'm allowed to cheat. Okay, we learned several more important things. One thing that we learned is that there are various possibilities depending on the values of h0 and j. So one possibility is that, let's consider this case of small j. For the case of small j, there's only one solution, only one possible solution, which is this point. So this is the only fixed point of this differential equation. On the other hand, in the case of a large j, we found three solutions. One solution, two solutions, second solution, and a third solution. Well, in the case of h, we had something that is very similar. So there was either possibility of one solution or three solutions, but it was very clear that if H will be even larger, then there will only be one solution. We'll call it even larger H0. So this tells us something. It tells us that the solution or the behavior of this differential equation is, well, qualitatively, or the qualitatively different regimes depending on the exact value of J and H0. There are some regimes in which we have only a single solution to this equation, like in this case, or there are regimes in which there are several solutions, three solutions. But still we are interested in how this network will behave. So what can we say? So we understand that the solution will. So we learned from this exercise is that the solution could depend in a qualitative way on the values of J and H0. This is one thing that we learned here. But still we don't know how this network will behave. So what should we do next? What should the next step be? Yes?
Try to see the eigenvalues, if they are positive, negative.
What do you mean by eigenvalues?
The Jacobi matrix.
I understand what you're saying, but I'd like you to be slightly more precise about the goal.
Okay, the goal is to determine the fixed points, if they are stable or unstable. If they are attracting fixed points or not attracting. So if everything converges to the certain fixed points or not.
Right. So the idea is the following. We already know that there is either one fixed point or several fixed points to these equations. This is one thing that we already know. But we don't know anything about the properties of these fixed points, whether these fixed points are stable or unstable, whether they are attractive or repulsive. And some of you have taken an entire course on this topic, but for those who haven't, I will, for the sake of those that haven't taken a 12 course, I will compute the stability of these fixed points. Who are the people who have taken 12 course, or who haven't? Or let me redefine my question. Are there people here who have never done, who have never analyzed the fixed point, the stability of a fixed point? Okay. So I will study the stability of this fixed point for this, or these fixed points for this particular example. So what is the idea? Actually, I think it's a good time to take a break, and after the break, I will analyze the stability of these fixed points.
You're not as fast.
No, I'm like,
Yeah,
but yeah,
but he was a good friend. He just that's all it was.
well, yeah.
Yeah.
Yeah, it's always fun to find your kids putting like that. It's just, and also, like, there's people there.
זהו, אז כאילו, נשארתי גם בכפר כזה, אבל לא באתי, עד שעכשיו קיבלתי את המייל
Yeah, okay.
שם קורס של
And no, it was
So,
great.
no, because like,
Yeah.
so like,
I have to do the
עכשיו קורס מתמטיקה שם, ככה ג'נטיקה.
that everybody
You have people
yeah, oh,
Okay, so we have this OTAPS model, and we know that depending on the parameters J and H0, drawing these curves, it seems that the solution has there are basically two possibilities: either that there will be one fixed point, or that there will be three fixed points. And but the fact that there is a fixed point doesn't mean that this is a solution in the sense that after a long period of time the dynamics will converge to a fixed point. The necessary, although not sufficient, a necessary condition for the dynamics to converge to a fixed point that it will be locally stable. And what we will do now is compute the local stability of the fixed point or fixed points of the dynamics. Now I know that many of you have done similar things, but still, again, today is like a introduction to biology, introduction to dynamics. So I will do it anyway. Now I'm going to assume that tau is equal to 1. This has no implication. Basically, we can think about measuring time in units of tau. So how do we study the stability of this fixed point? So the idea is that we assume that H at some point in time is close to the fixed point, which we denote by H star. So H is equal to H star plus DH, where H star is such that minus H star plus J G H star plus H0 is equal to 0. So this is a fixed point equation, and we assume that H is not too far from this fixed point equation. And now we look at the dynamics of delta H. And the way that we do it is by taking the temporal derivative of H. So H star plus DH dot, where this dot denotes a time derivative, is equal to minus H. So it's equal to minus H star plus DH plus J times G of H star plus DH plus H0. So far we haven't done anything. We just measured H relative to H star, and we wrote down this differential equation. So the next step would be, well, the temporal derivative of H star plus DH is just equal to DH dot. Let's look at the right-hand side of the equation. What we're going to do, we are going to Taylor expand this G function. So we'll have minus H star plus DH plus J times G H star plus J G prime H star times DH plus H0. So what we did here is to Taylor expand this G function around H star. The next step would be to realize that minus H star plus JGH star plus H0 is equal to 0 because H star is a fixed point. So this is just equal to minus delta H plus J G prime H star times delta H, or rewriting it, delta H dot is just equal to minus 1 minus j g prime h star times delta h, or solving this differential equation, delta h is equal to delta h0 times e to the minus lambda t, where lambda is just equal to 1 minus j times g prime h star. Questions about this? So what are the conditions for stability of the fixed point? When will the fixed point be stable? So if 1 minus j g prime of h star is positive, if lambda is positive, then, so let's consider the case of lambda. There will be two possibilities, either, or there are three possibilities, but two interesting possibilities, either that g is positive or that, either that lambda is positive or that lambda is negative. Let's consider first a situation where lambda is positive. If lambda is positive, this implies that if we start close to or near a fixed point, if we start near a fixed point, then the distance from the fixed point, delta h, the magnitude of the distance from the fixed point, will decrease exponentially fast. So if we start from a region that is close to a fixed point, then the dynamics will converge to this fixed point. On the other hand, if lambda is smaller than 0, if we start near a fixed point, then the distance from the fixed point will diverge. So a condition for stability of the fixed point is that lambda is positive, whereas the condition for instability is that lambda is negative. Questions about this? So there are several questions here. First question is, can we say something more interesting than just talk about, so let's say that we understand this. Let's say that we found that the fixed point is positive, that the lambda is positive or lambda is negative, and we understand what's going on near the fixed point. Can we say something more intelligent or more comprehensive, perhaps, about what's going on for all values of h? That's the first question. Second question is, what would happen if lambda is equal to 0? It's also perhaps an interesting question. So regarding the first question of what can we say about solutions that are outside or far away, or initial conditions that are far from the fixed point, this is something that we will go back to at a later stage. We will discuss it later. What about the case of lambda being equal to 0? What should we do? What can we say about, if lambda is equal to 0, what can we say about the fixed point?
Can be either stable or not stable depending on the next stage in the Taylor expansion. But in any case, in this case, the time scale which will take it to change value is much smaller.
So you're right in saying that if lambda is equal to 0, then in order to address the question of stability of the fixed point, we should discuss the higher order terms in this Taylor expansion. So a nice way of thinking about it or remembering it is to consider the following differential equation. X dot is equal to minus x to the power of 3. So what are the fixed points of this dynamics? Zero. Okay. So the fixed point is 0. If we Taylor expand this term around 0, what would be the eigenvalue, if you like, of the matrix, or what would be the value of lambda here? What's the Taylor expansion of this? Around 0? I'm sorry?
Minus 6 to the third. First order is 0.
First order is 0. And so if we just use this method, we'll be unable to say whether it's stable or unstable, similar to the case where we have this equation, x dot is equal to x to the power of 3. But we know that for this equation, when x is positive, then x dot is negative. So we convert, we go in the direction of going towards 0. When x is negative, x dot is positive. So again, we go in the direction of 0. So this is, in this case, the 0 fixed point is a stable fixed point. Whereas in this case, the 0 fixed point is an unstable fixed point. And we cannot use this method, taking the first linearizing this x cube function in order to determine stability. But we will not worry about it at the moment. Okay, so now let's go back to our drawing of this function, or finding the fixed points. And my question to you is, which of the fixed points are stable and which ones are unstable, and why? Positive. J is positive. So can we say something about the stability of the fixed points without being more precise about the exact shape of G or about the exact values of H0 and of J? Examine the slope. Okay. Which fixed point would you like to consider? Choose a fixed point. This one. So we are considering this fixed point. So what can you say about this fixed point? Is it stable or is it unstable? And why? Why is it stable?
The slope of J
So in order for an intersection between J, G, H plus H0, an intersection of this function with H is equal to H, in order for this intersection to take place, the slope of this function has to be smaller than the slope of this function. So what is the slope of this function at this point? Well, the slope is simply J times G prime. So we know that at this point, this slope is smaller than 1. So if this slope is smaller than 1, well, this implies that lambda is positive. So even without going into the details of G and J and everything, we already know that this should be a stable fixed point. Just by the fact that in order for intersection to take place, the slope of this green function should be smaller than 1. Is this point clear? Yes?
Why, if J is, so why do you know, why is J so, let's say, so, like, bigger than large enough? What's the argument that J is large enough? That's lambda. Lambda should be positive, right? To have a stable fixed point.
It goes the other way around. I mean, we consider the situation where J is large enough so that we will have 3 intersections. We have 3 fixed points. Now we consider this fixed point. Now we know that in the limit, the slope of this goes to 0. So in order for you to have 3 intersections, the slope of this function here should be smaller than 1 at this intersection. If the slope of this function at this interaction is smaller than 1, well, this implies that G prime at the fixed point is smaller than 1. Now, if G prime at the fixed point is smaller than 1, then lambda necessarily is positive. So this implies that the fixed point has to be stable. Similarly, what can we say about this fixed point? Well, it's exactly the same thing. Here, the slope is also smaller than 1. So if the slope is smaller than 1, well, again, lambda is positive. What about this fixed point? This intermediate fixed point? Well, here the slope is larger than 1. So necessarily, lambda is smaller than 0. So this is an unstable fixed point, whereas these 2 extreme fixed points have to be stable. What about this case? There are too many. Let's throw it again. So, what about this boring case, where we have only 1 fixed point? Is it stable or unstable in the case of a single fixed point? Well, using the same argumentation, we know that the slope here has to be smaller than 1. If the slope would have been larger than 1, then at some point there would have been another fixed point. So we know that, that for the case of a single fixed point, the slope is smaller than 1, and as a result we know that the single fixed point is stable. Put differently, looking at this model of the OTAPs, there are 2 solutions that are qualitatively different. One solution is a single stable fixed point, and the other solution are 3 fixed points in which the 2 extreme fixed points are also stable, and the intermediate fixed point is unstable. And this we can conclude without saying anything more precise about the function g and the exact values of h0 and j than what we did here. Basically, g has to be a sigmoidal function, a monotonic increasing function with saturation. Questions? So one thing that is perhaps of interest that we have learned from the OTAPs is the fact that network interactions, well, this self-excitation, turned a single neuron that had a memory of tau that converged, a neuron that forgets the initial conditions exponentially fast with a time scale of tau, to a system that is potentially bistable. And a bistable system, in principle, can store memory of its state forever. So from a computational point of view, this network interaction is a first step towards long-term memory. So we take a system whose characteristic timescale is tau, but in principle, if J is sufficiently large, and if we are in this regime of bistability, can store some information about the past forever. Questions about this network before moving on to another example?
What do you mean when you say that the system remembers, that has memory?
Well, there are two stable fixed points. So if you start from near one fixed point, you'll converge to this fixed point and not to the other fixed point. If you start near the other fixed point, you'll converge to this fixed point and not to the first fixed point. So in principle, this system remembers the initial conditions forever. Not everything about the initial conditions, one bit perhaps of memory about, one bit of information about the initial conditions. Okay, let's take a real network now. Let's do something more interesting. Okay, example 3. So this example is a network with all-to-all connectivity. Let's write down the equations. So SI is equal to G of HI. And we have tau HI dot is equal to minus HI plus sum over J of Jij Sj plus HI 0. And we will assume that all neurons are connected. Jij is equal to some J over N. And for simplicity, as before, we will assume that tau is equal to 1. So let's remove this tau. So how should we go about analyzing this network? Yes? Yeah.
Just one constant.
Yes. Where N is the number of neurons in the network. So how should we analyze this network? We have a set of differential equations. Previously we had one differential equation. Now we have n differential equations. What should we do?
First of all, there's
I'm sorry?
The symmetry in the equation.
The symmetry in the equations, okay.
Whatever fixed point you find, you can. There are many others that are basically the same.
Well, there's symmetry in the equation. Is it clear that there's symmetry in the solution?
Because
Why wouldn't it be? That's an interesting question, and we will leave it. Well, so the short answer is that in this example, the equations are symmetric, and the solution would be symmetric. And in a second, we will see that you're right. But this is not necessarily true in general. And in the next example, we will see that it's not.
But the starting point won't be symmetric for something. Something has to be asymmetric for a symmetric system to produce a non-symmetric.
Well, we will see. We will see in the next example. So let's write again. Let's do what you suggest. Introduce these. Incorporate these J.
Jonathan, did you say something about H0?
No, haven't. I will say it in a second. Yeah, we will assume that H0 is equal to. We'll assume symmetry. That we always see the same input. It's a good point. Thank you. So I'm writing the equation hi dot is equal to minus hi plus j over n sum over j of g hj plus h0. So the fixed point solution is just hi is equal to j over n sum over j g of hj plus h0. So following your suggestion, we see that the fixed point solution is the same for all neurons. So what would be the fixed point solution here? So the fixed point solution will be hi is equal to some h. And we have the equation that h is just equal to j times g of h plus h0. So this is a fixed point solution. Familiar? So this is exactly the same fixed point solution as in the case of the OTAPs, which we already know well and like very much. So the fixed point solution in the case of all-to-all connectivity with, as Itamar correctly noted, with equal inputs to all neurons is exactly the fixed point solution of the OTAP differential equation. What should be the next step? Or are we done? So we need to do stability analysis. Is it exactly the same? What's the difference? How do we do stability analysis here?
It's just more dimension.
Okay, so how do we do it? Let's do stability analysis. So how do we do stability analysis? Yes?
We could do a Taylor expansion just with more dimensions. Like similar.
Because if all solutions are similar, and you can just do in one case, then all the other cases will be the same. Or am I wrong?
Can you be more precise?
If one neuron reaches a certain fixed point, we know that all other neurons behave the same. So all the other neurons will have exactly the same fixed point with the exact flexibility.
Well, we don't know that they will reach a fixed point. We know that this is a fixed point. Maybe the dynamics is more complicated. So in the one-dimensional case, you could use other forms. Well, okay, cutting a long story short, the answer would be we will get the same, the analysis of stability for this all-to-all network would be the same or would result in the same result as in the case of the one neuron. But this is not a trivial thing, because we know some things about one-dimensional systems that are no longer true for multidimensional systems. So we will do it anyway, and perhaps in the next example perhaps would be more interesting for the two of you concerning your question about the relation of symmetry in equation and symmetry of solution. So let's do stability analysis. And it's also a nice exercise in the sense that for those that have never done stability analysis in a multidimensional system, this is a time to study it. So how do we do stability analysis? The same as before. Basically what we do stability analysis. And we do it in exactly the same way as before. So we assume that the inputs to the different neurons is equal to the fixed point value plus some perturbation, and that the fixed point value is just H star is equal to J, G, H star plus H0. As before. And now we take these equations. And as before, we have H star plus delta Hi dot is equal to minus H star plus delta Hi. This term plus J over N sum over J of G of H star plus delta H. J plus H0 and as before we Taylor expand this G function resulting in delta Hi dot is equal to minus delta Hi plus J over N sum over J G prime at H star times delta Hj. And we're interested in solving, this is a set of linear differential equations, and we're interested in solving them. So how do we go about doing it? Let's remind ourselves the general case. So let's say so how do we solve this this equation? So this is a set of linear differential equations in matrix form. Basically, this is a generalization of this equation. So what do we do? So if the matrix M is diagonalizable over the complex numbers, what we can do, we can write delta H to be, we can expand it using the eigenvectors of the matrix M. Right? This is a reminder to all of you. Now I will add, I will just for clarity, we'll think about UJs as the right eigenvectors of the matrix M. So I will add R, just to remind you that we're talking about the right eigenvectors of M. And then we have delta H dot is just equal to sum over J of AJ dot UJR, which is equal to M times delta H, which is sum over J of AJ UJR, which is just equal to sum over J of AJ. I'm taking M inside, and MU is just equal to lambda U. So I have AJ lambda J UJ R. So what's the next step? So I'm going to multiply this equation. I'm going to multiply this equation from the left with the left eigenvectors of M. And I'm using the knowing that UI left times UJR is equal to delta IJ. So I'm multiplying this equation from the left with the left eigenvector of the matrix M. So I'll multiply it by the kth left eigenvector of M. So from this left-hand side of the equation, all that is left is just AK dot. And from this side of the equation, all that is left, again, is AK times lambda K. Or AK T is equal to AK 0 e to the lambda kt. So this is a way of turning this basically separating the different terms in this, or the different variables in this equation. So what is the condition for stability of fixed point according to this analysis? So the fixed point would be stable if all these eigenvalues of the matrix M are smaller than 0. So there are 2 points noteworthy. First, the way that we defined lambda previously in the one-dimensional case was the opposite. We had a minus here, and therefore all the everything that we said there is opposite here. Is it an if and only if condition? Is it an if and only if condition?
It can be also equal to 0.
And then,
It can be a saddle point.
if it's equal to 0, is it stable or unstable? We don't know. So it's not an if and only if. But if all eigenvalues are negative, then we know that it's locally stable. If some are equal to, if one is positive, if at least one is positive, then we know that it's unstable. If they're equal to 0, then we need to take the higher order terms. So in order to do a stability analysis to this case, to the all-to-all connectivity, basically what we need to do is look at this equation, extract the stability matrix, the Jacobian, and compute its eigenvalues. And then look at the sign of these eigenvalues. So let's look at this set of equations, and let's look at the stability matrix. So how does this matrix look like, the matrix M? So what would be the contribution of this term to the matrix M? Delta HI dot is equal to minus delta HI. So how does the matrix look like? Yeah, minus the identity matrix. Good. Okay, let's look at this one. Well, we have J over plus, that's easy. J over N, that's easy, times G prime, which is common to all. So for every element we have contribution of sum of all other elements. So basically it's a matrix that looks like this. matrix of ones. Right? This is for delta H1, sum over all of them. This is for delta H2, sum over all. This is for delta Hn, sum over all. Or one way of writing it would be minus I plus j over n, g prime of h times, if we like MATLAB, I would call it 1 times 1 transpose. So this is a vector of 1s times, well, another vector of 1s. A column vector of 1 times a row vector of 1s. Okay. After some effort, we found this stability matrix M. Now we need to compute the eigenvalues of this matrix. What do we do? Yes. Okay, so characteristic polynomial is the standard answer.